Okay, so welcome to another video where we're going to discuss some biology questions. In this case, we're going to discuss questions in molecular biology and genetics. So let's start. So before we go in, you have to be aware that this paper's got, I think, three sections. There is section A, B, and C. So section A has got 25 questions. And as you can see here, it is indicated that a total marks for this section is 25. That is, each single question carries a single mark. So let's start. Question 1. The Messier-Stone and the star experiment demonstrated that so as we know, the Messier-Stone and the star experiment, this is the most important experiment in as far as molecular biology is concerned. So this is an experiment which was conducted by these two scientists, Matthews Messier-Stone and Franklin Starr. In the year they conducted this experiment somewhere in the year 1957 to 1958, somewhere there. So this experiment was conducted to confirm which of the three models of DNA replication is true. Because at first DNA replication was being considered to be in three different models. Like DNA replication takes place in three different models. And these models were that DNA replication is dispersive, that is, each strand contains both new and old strand. Then others used to look at it that DNA replication is conservative, that is, the new DNA will contain 100% old strand, then the other one is going to contain 100% both new strand. Then others used to look at it that DNA replication is same conservative, that is, each newly synthesized DNA is going to have half old strand and half new strand. That is, if this is the old strand, this is going to be the new strand. So this is the same conservative. And this experiment, it confirmed to say that DNA replication is a semi-conservative. That is, each newly synthesized DNA is going to have one strand which will be old, and the other strand, the new strand, is going to be new. So why is it so? It's because when we say half old strand and half new strand, it's like this. You know, to say that DNA replication is a double-stranded molecule, like this. So if this is 3 prime, this will be 5 prime, this will be 3 prime, that will be 5 prime. So when this DNA replicates, this strand, these two strands are going to separate, of course. So we're going to have these two odd strands. Okay, this will be odd strand. Let's say this is 5 to 3, this is 3 to 5. Then from there we are going to have a new strand. So a new strand is going to be like that. So I'm using a different color to show that this is going to be a new strand. So this is going to be 3 prime end, that will be 5 prime end, this will be 5, that will be 3 prime end. So this is what the experiment of Messier-Stone and Starr confirmed, that DNA replication is a semi-conservative. So the answer here has to be option D. A is saying DNA is a double-stranded structure. This is not what the experiment was about. B is saying DNA replicates non-conservatively. As I've already explained, it replicates semi-conservatively. Then C is saying DNA replicates conservatively. So conservative, what it means is that these new strands of DNA are going to have, let's say, this one will have both old strand, then this one will have both new strand, which is not true. So each one of the newly synthesized DNA each one of them is going to have one old strand and one new strand. That is semi-conservative. So the answer is D, which is saying DNA replicates semi-conservative. That is what this experiment was all about. So we move on to question 2. So question 2 reads, the template strand used during transcription ran in the template strand. So you know to say that DNA has two strands, right? So these two strands have got a different direction. We have the 5 prime end, this will be 3 prime end, then automatically this will be 3 prime end, that will be 5 prime end. Okay, so you have got a DNA like this. So between these two strands, one of them is going to act as a template. A template is a guide to the messenger RNA. So you know to say that messenger RNA runs in which direction. The messenger RNA is always synthesized or transcribed in the 5 to 3 direction. So the template strand of DNA has to be opposite to this direction. That is, the template strand of DNA has to be in the 3' end to 5' end. That is the template of DNA, the one which runs in the 3' end to 5' end. So the answer here has to be option A. That is the template strand. As you know that the template strand has got a lot of names: the non-coding strand, the antisense strand, and the template strand. All these names refer to one and the same strand, which runs in the 3 to 5 direction, whereas the other one is the sense strand, which runs in the 5 to 3 direction. Okay, so the answer there is going to be A. We move on to question 3. Question 3 reads, which enzyme is involved in the excision repair of thymine dimers? So thymine dimers, you know to say, so this is much about DNA. So we have got these two strands. So a dimer, let's say you have thymine there, then adenine, another thymine, then cytosine, guanine, say adenine. So I'll only focus on a single strand. A thymine dimer is when two adjacent thymine bases react together. Okay, so these are adjacent what? Adjacent thymine. So when these thymine, they form more like a bond on the same strand. That is what is known as a thymine dimer, and that is going to result into a mutation. So we have got enzymes known as excision repair. These enzymes, what they do is that they will cut this segment which has got a dimer. So they will cut there and here, then remove it. When they remove it, they are going to join the remaining what? The remaining pairs. So they will remove that dimer to prevent further mutations. But of course, the removal of this dimer is going to result into a mutation. But these excision repair enzymes, this is what they do. So the question is, which enzymes is involved in the excision repair, in the removal of these thymine dimers? So we have got fertilize, that is A. 2, we have got endonuclease. C, we have got DNA polymerase 3. So DNA polymerase 3, this is involved in proofreading. So this one is just going to proofread. That is, it checks that if the bases here are normal paired. This one has to be paired to guanine, cytosine, thymine. That is what this O does. It does not cleave, or it cannot cut these where these dimers are formed. Then we have telomerase. So telomerase, these are enzymes which will only cut at the tip or at the end of the base. So these dimers can form within the strand of DNA. So that can be within. So the enzymes which are involved in the excision repair of thymine dimers are what we call endonuclease enzymes. So endonuclease, endo simply means inside. We also have got exonuclease. So exonuclease, these are the ones which are involved in the proofreading of DNA polymerase 3 and 1. Okay, so these will only cut at the end. Okay, they will check the base pairing here. If it's not, the DNA are not correctly paired, the bases are not correctly paired, it will cut just here. It cannot cut within. The only system that has got that ability to cut the bases within this DNA strand are what we call endonuclease. Those enzymes which have got endonuclease activity. So the answer there is option B for question 3. We move on to question 4. So question 4 is, which of the following traits is an example of dominant sex-linked trait? So dominant sex-linked trait. So here there are two terms which you have to understand. The first one is sex-linked. What does it mean when you say sex-linked? So when we say that a particular trait or disease is sex-linked, that means it's only carried by the what? The X chromosome. Okay? It can only be carried by this X chromosome. Those mutations which are carried by the X chromosome are the ones which we refer to as sex-linked or X-linked disorders. So the question here is saying which of the following diseases is an example of dominant sex-linked trait. So you also have to understand it can be X-linked, but is it dominant or recessive? So dominant, that means that trait is inherited in a dominant form, okay? Whereas if we say it's recessive, it means that it's inherited in the recessive form. So I've got a number of disorders or traits which are inherited as in a recessive form. We have a few which are inherited in as a dominant trait. An example is Fragile X syndrome. Fragile X syndrome is inherited as a dominant sex-linked disorder. So which one of the following is an example of a trait which is inherited as a dominant linked trait? So A, we have sickle cell anemia. So sickle cell anemia is an example of autosomal recessive. So it's inherited as autosomal recessive. So autosomal simply means that that particular trait or that particular disease is carried on any of the chromosome from chromosome 1 to 22. Any of the disease or trait that is carried on the chromosome 1 up to 22, that disease is referred to as an autosomal disease or trait. Whereas the 23rd pair, which is a sex-determining gene that contains, if it's in males, you have got XY. If it's in females, you have got XX. So, disorders which are carried on the X chromosome, that is what is known as sex-linked trait or disorder. So, sickle cell anemia is an example of autosomal recessive. Huntington disease, this is an example of autosomal dominant. So it's inherited in a dominant form, like that. Then phenylketonuria. Phenylketonuria, this is also an autosomal recessive, disorder, if I'm not mistaken. Then we have hemophilia. So hemophilia, this is an X-linked recessive. So it's an X-linked recessive. I said that X-linked and sex-linked is just one and the same thing. So all the options given here, the question is demanding us to come up with a trait or to identify a trait which is inherited as a dominant sex-linked trait. So from the options given, we don't have a dominant sex-linked. We only have a single sex-linked trait, which is hemophilia, but however, this hemophilia is not a dominant trait. So as far as our options are concerned here, we don't have a correct option to this question. We only have one sex-linked recessive trait. So there was no answer to this question. Okay? There was no answer to this question. I'm sure corrections were made during marking or maybe during an exam. Corrections, I'm sure, were made. Question 5. Down syndrome is a result of trisomy at chromosome number. So Down syndrome is due to mutation. There's an added extra chromosome to chromosome 21. We know that these chromosomes, they occur in pairs. That is, each chromosome has got a pair of chromosomes. But for trisomy, instead of having a pair, you have got 3 chromosomes at chromosome 21. So that is Down syndrome. It's also referred to as trisomy 21 because it comes as a result of mutation on chromosome 21. Question 6. Which of the following statements best describes a term penetrance? So penetrance, this is the likelihood or the percentage of an individual carrying a particular trait or particular genotype to express that particular phenotype. It is the likelihood or the percentage of an individual carrying a specific genotype to express a particular phenotype. You know that the phenotype is detected by the genotype that a particular individual carries. So the percentage of the population, let's say out of 100%, these individuals carrying different genotypes, the likelihood of total number of individuals who can express that phenotype, that is what is referred to as penetrance. So penetrance is important in a way that it can help us to predict the number of individuals who are going to express that particular phenotype, that are going to express particular phenotype having a particular genotype. So that is what penetrance is all about. And a good example of penetrance is breast cancer, where you have got a gene BRCA1 and BRCA2. So depending on the genotype that a particular individual is carrying, that is going to be expressed. Some is going to be expressed in their phenotype, some is not going to be expressed. So that is what penetrance is all about. So it's looking at the likelihood of an individual carrying a particular genotype to express the phenotype. So A is saying the modifying effect on the environment and the genotype on a particular phenotype. So no. B is saying the degree of effect produced by a penetrant genotype. No. Then the masking effect of one pair of arrow by another pair, so that is epistasis. Then 6 is saying the proportion of individual with a particular gene combination which show a corresponding genotype. The proportion, so this can be the percentage of an individual with a particular gene combination which show a corresponding phenotype. So that's the answer, D. So that is just the proportion or the percentage of an individual with a particular gene combination which show a corresponding phenotype. So D for question 6. 7. During base analog mutation, ionized form of 5-bromouracil will pair with which base? So 5-bromouracil, this is a mutated, it's a uracil, but it mimics thymine. Okay, it mimics thymine. So what will happen is that when this mutation occurs, it's a uracil, but it has got the characteristics of thymine. So you know that thymine will pair with what? Adenine. So instead of it pairing with adenine, it pairs with guanine. So there's that mutation. There's that base mismatch. So 5-bromouracil, it pairs with what? Guanine. It mimics thymine, but instead of it pairing with adenine, it pairs with guanine. So the answer here is it has to be so the answer there has to be adenine. So it pairs with adenine. So question 7, which reads, during base analog mutation, ionized form of 5-bromouracil will pair with which base? So 5-bromouracil, this is a mutated uracil. So it mimics the activities of thymine. So it will not act as uracil as per se. Instead, it's going to mutate or to mimic the activities of thymine. So because it has got these activities of thymine, it's going to pair with, we know that thymine will pair with what? Adenine. So the answer there has to be D, adenine. That is, 5-bromouracil will mimic the activities of thymine, thereby pairing with adenine. That is going to be a false pair. Okay, it's a mutation. Question 8. Addition of a 5' cap and a 3-pore tail is an example of. So we know that the capping and pore generation, we only see these in translation or in transcription. We only see this in transcription. Capping is the addition of a modified guanine, a 7-methyl guanosine molecule, which is added at the 5' end. Then the pore tail, this is an addition of a repeated unit of adenine molecule at the 3' end, and that is why it's known as a pore tail. So the question is saying that these two are examples of a transcriptional control. So A can be true because they are associated with transcription. Let's check these other options. B is saying translational control, so it doesn't deal with translational control. post-transcriptional control. So post-transcriptional control will be disqualified because capping takes place as soon as mRNA is synthesized. As soon as mRNA starts to be synthesized, the 5' end has to be protected from degradation. So capping will take place here to prevent this 5' end to be degraded by enzymes or the cell. So it's not part of the post-transcriptional control, okay, but it's part of the transcriptional control. Then what comprises the post-transcriptional control is splicing, whereby now you remove the non-coding regions, which are introns. Then you leave the coding regions, which are exons. That is what makes up the post-transcriptional control. But these two are just part of the transcription control. That is capping and pore regeneration. So these same post-transcription control. So as we have already said, these are associated with transcription. So the answer there is A for question 8. Then question 9. Question 9 reads, groundnuts plants produce paper flower when both dominant gene, that is C and P, are present in the genotype, and produce white flowers when either or both are absent. Determine the genotypic ratio from this cross. So I've got this cross. So that is okay, so let's do this. So the key here is that these two, both dominant C and dominant P, they have to be present for that particular plant to have what? Paper flowers. Then if in either of these is absent, that particular plant is going to have what? White flowers. So let's conduct or let's do a cross so that we see what we're going to get from there. So of course this is going to be a Punnett square. It's going to lead us into a Punnett square. So let's do this, though it will take a bit of some time. So we have let's first okay, let's start with the gamete. So that is the cross between we have we have this and that, then this and this being crossed with this and that. So first of all, you know that this is a dihybrid cross, so you first have to come up with the gametes. So I'll start the gametes of this one. This one will give us 4 gametes. So that is going to be capital letter C, capital letter P. That is this and that. Then the other one is going to be capital letter C, small letter P. So we have this and that, because it's going like that now. So we have done this. So we have 2. Next, this one has to pair with this also and that to complete the 4 gametes from there. So we have small letter, this and that, and that. Then I'll come to this, the same has to repeat. So another set of gametes is going to be small letter, this and that, then this and that. Then, okay, this is what we have. So we can now come up with a Punnett square. So a Punnett square, you know that this is going to give us how many? It will give us 16 offsprings. So all the 16, then we have now to analyze which one of those are going to have both capital letter C and the small letter or capital letter P, which are going to be purple in color. Then others are going to, those which are going to lack any of the dominant either P or C are going to be white in color. So that is what we want to analyze. So you can't answer this question minus doing this, minus conducting the Punnett square, minus using this Punnett square. Because it is a dihybrid cross, so you just have to use a Punnett square. That is the only way on how you are going to answer that particular question. So I want you to follow very close here, because this is an important tool in molecular biology, Punnett square. Then you are going to feed your gametes. So we'll start. So we are going to have, say, female gametes, then male gametes, though it doesn't matter. So we'll start here. I'll start with these gametes. I'll feed them in here, one in each. So I'll start with this and that, this and that, then we have this and that, and then finally we have that. The same here. So these are small letters, that, then this, then another one, then this. Then now we have to conduct a cross. So we are going to have this will be this, that, automatically that is going to be purple. Then you have this, and and the small letter p. Then this will be small letter c, p, p. Then c, c, p, p. Then I'll come here, capital letter C, small letter this, capital letter P, small letter p. Then capital letter C, small letter c, small letter p, small letter p. Then small letter c, c, capital letter P, small letter p. Then this. Hope you are able to follow where these are coming from. So this, then that, another one. Then we have another one here, this, that, then this, and that, then this, and that. So this is capital letter P is coming from there, small letter p is coming from here. So that's what we have there. Then the last column we have capital letter C, small letter c, capital letter P, small letter p. Then capital letter C, small letter c, small letter p, small letter p. Then C, c, capital letter P, small letter p. Then we have C, c, p, p, small letter p's. So now we have to count. The statement here was saying the groundnuts produce purple flower when both dominant genes C and P are present in the genotype, and produce white flowers when either or both are absent. Determine the genotype, the genotypic ratio from this cross. Okay, so we now have to start counting. How many are going to have both capital letter C and capital letter P in them? All those which are going to have both dominant C and dominant P gene, they are going to be what? They are going to be purple. So we start counting. 1, 2, 3, then we have 4, 5, 6. So we have 6. Okay, 6, 2. The rest are going to be what? The rest now are going to be purple, or they are going to be white, because they like either C or either capital letter P or both of them. As for this one, it doesn't have capital letter C, so it's going to be automatically white. The same with this one. This one likes capital letter P, this one likes capital letter C, this one likes capital letter both of them. Then this one likes capital letter C, this likes capital letter C as well. This one likes capital letter P, this one likes capital letter C, this one also likes capital letter C. So you know that we have 16 total progenes. So 6 of them are going to have that gene, they are going to be purple, then the remaining 10 are going to be white. So if we reduce these ratios, we are going to divide both by 6 by 2, that will be 3, this one will be 5. So the answer has to be 3 to 5. That is going to be the phenotypic ratio, 3 to 5. So 3 are going to be purple, 5 are going to be white. As you can see there, our answer is going to be option C. So our answer is option C. Hence we have answered this question. So that's how simple you can answer these questions involving Punnett squared. Question 10. Which of the following scenarios will turn on the lycopene? So we know that the lycopene will only be turned on in the presence of lactose, lactose has to be present, then glucose has to be what? Glucose has to be absent, or these have to be in higher levels than this one. This is the situation in which the lac operon is going to be on. So A is saying glucose and lactose are present. So if both are present, the bacteria is going to utilize glucose. It's not going to utilize lactose. So when the bacteria is not utilizing lactose, the lac operon is going to be off because it does not need those enzymes to break down lactose into its simplest sugar. So it's just going to use glucose because glucose is the simplest form of a sugar. Okay, so it will be off in option A. B is saying glucose and lactose absent. So if both are absent, it's going to be off. Okay. C is going glucose absent and lactose present. So glucose has to be absent and lactose has to be present. In this case, it's going to be on. Then glucose present and lactose are absent. So in this case, it will be off. So A, B, and C, the lac operon is going to be off. Only C is the only condition in which the lac operon is going to be on. Because in this case, when glucose is absent, and lactose is present, the bacteria has to transcribe those genes which can metabolize or break down this lactose into its simplest sugars, that is glucose and galactose. Then now this bacteria can utilize what? Glucose. But if glucose is present, there's no need of this bacteria to transcribe those genes to break down this lactose because glucose is already available. So glucose has to be absent, lactose has to be present. That is the condition in which the lac operon is going to be on. So option C there. 11 reads, which of the genotype below represents a sperm that can unite with a normal egg to produce a zygote that will develop into a normal human male embryo? So this question has to do with you understanding the genotypes of male and females. You know to say that males have got XY. Any individual who carries the XY chromosome, that individual is male. Whereas females have got XX. So we know that the first 22 pairs of a chromosome, those pairs are known as autosomal chromosomes. So we're going to say you have 22 autosomal chromosomes. Then to complete, because you know that each and every human being has got 23 pairs of chromosomes. So now we are saying the 23 pairs of chromosomes, 22 of those are what? Autosomal chromosomes. So we have 22 autosomal chromosomes. For this individual to be male, the 23rd pair has to be XY. If you put XX as the 23rd pair, that particular individual is going to be what? Female. So the question is saying which of these genotypes represents a sperm that will unite with a normal egg to produce a zygote that will develop into a normal human male embryo. So if you have got XY, this one when it unites with an egg has got XX. In this case, you have a chance of producing a male. This is going to be male. Then that one will also be what? Male. Here you have a chance of producing a male. Okay? But if you say XX, it means that you are uniting an egg with its own fellow egg. In short, two females cannot produce an embryo. So the answer there has to be 22 autosomes and XY. So A. This one we don't have such. This one also we don't have such, and that one. So it has to be a pair of chromosomes. So C and D, they are not pairs of chromosomes. So the answer here is C, is A. The A that you see in front of 22 is simply representing autosomal. That is the autosomal chromosomes. That's what it means. Then 12. Erythroblastosis fetalis is in pregnant women occurs when. So this is a hemolytic disease of a newborn. Erythroblastosis fetalis. So this one has to do with the rhesus factor. So what happens? So in rhesus factor, let's say you have got a couple, you have got a male and female. So an individual who has got who is having a rhesus factor, that individual is regarded as a rhesus positive. Okay, rhesus positive. An individual who doesn't have a rhesus factor on the cell surface of the red blood cell, that individual is regarded as a rhesus negative. So the baby takes up the rhesus factor of the father. If the father is rhesus positive, the baby is also going to be rhesus positive. If the father is rhesus negative, the baby is also going to be rhesus negative. Okay? So this situation is a situation where during delivery, if there is a mix-up of blood, let's say a baby is rhesus positive, and the mother is rhesus negative. In this case, this is a baby, okay? This is a mother. So if during delivery there is a mix-up of blood between this baby and the mother, the cells of the blood cells of the mother are going to be sensitized. They are going to be sensitized. That is, they are going to produce antibodies against rhesus positive. So if this mother is to have a second child, and if it so happens that that second child is rhesus positive, the blood of the mother has already been sensitized against rhesus positive. So they are going to produce antibodies. The antibodies of the mother are going now to react against the rhesus positive fetus. And this fetus is going to die. This is going to be a premature death of the fetus. That is what we call erythroblastosis fetalis, also known as hemolytic disease of the newborn. Okay? So rhesus factor, if the mother is rhesus positive, well and good. In this case, if the baby is rhesus negative, nothing is going to happen. Even if the blood mix up, nothing is going to happen. But it only becomes bad if the mother is rhesus negative and the baby is rhesus positive. Then during delivery, the blood of the fetus and the mother mixes up. The first child is going to be delivered. The first child will not die. But if the mother is now to carry the second pregnant, the baby is going to die because the blood of the mother has been sensitized against rhesus positive. So it's going to produce antibodies which are going to react against what? Rhesus positive fetus, thereby attacking it, and it's going to die a premature death. So erythroblastosis fetalis in pregnant women occurs when so the husband has to be rhesus positive, because if the husband is rhesus positive, we know that the baby is also going to be what? Rhesus positive. Then the mother has to be rhesus negative. In this situation, that's when there's going to be erythroblastosis fetalis. So the husband is rhesus negative, no. Husband rhesus positive, wife rhesus negative, and offspring rhesus positive. So this is what I've explained here. So that is the situation in which now this erythroblastosis fetalis in a pregnant woman will occur. There's going to be the blood of the mother is going to be sensitized against the rhesus positive blood of the fetus. Then she's saying rhesus positive, husband has to be negative, husband positive, mother negative. So the baby and the father have to be rhesus positive, the fetus and the father have to be rhesus positive, and the mother has to be rhesus negative. That is a situation where you're going to have that erythroblastosis fetalis, the premature death of the fetus. 13. Given that black is dominant to brown, what ratio do you expect in the F1 hybrid close with the homozygous recessive brown parent? So let's say we are saying this is black is dominant over brown. Okay, so this will be brown. So it's saying, given that black is dominant over brown, what ratio do you expect in the F1 hybrid if the F1 hybrid is crossed with a homozygous recessive brown parent? So in this case, we are going to have all the F1 are going to be homozygous, heterozygous dominant, heterozygous black. So we are saying now we cross this with a homozygous recessive brown, like this. So what ratio? What would you expect? So here now we're going to have this and that. I'm doing shortcuts here. Then this and that, we have this. Then this and that, we have that. Then these two are going to give us this. So in this case, we're going to have 2, which will be black. 2 will be brown. So this is a 1-to-1 ratio. So the answer has to be option D. So that's the answer to question 13. And that's how you can answer this question. Okay, so you can use cross diagrams or a Punnett square because it's a monohybrid cross. 14. The cross in the question 13 above is an example of, so we had this from the F1, then we cross it with a recessive brown. So this is an example of a test cross. This is an example of a test cross. A test cross is used to determine the genotype or the dominant trait. You know, to say that if in the F1, okay, you had parents, black being crossed with this. From this cross, all the offsprings, you are going to have 4 offsprings here. All of them are going to be black. But you cross one black and one brown. So why is it that all the F1 are what? All the F1 are black. So in this case, you don't know the genotype of the F1. So you're going to conduct a test cross. That is, you get one of the F1 offsprings and cross it with the parent that has got a dominantly recessive trait. So in this case, the dominant recessive trait is going to, or homozygous recessive trait, the homozygous recessive trait is going to be this individual. So you get one F1 and cross it with the homozygous recessive trait. That is a test cross, so that you determine the genotype of the F1. The genotype of the F1. And when you conduct that cross, you are going to have wait, what did we say on question 13 again? So the answer on question 14 is test cross. So question 13, what did we say first? Given that the black is dominant to brown, what ratio would you expect if the F1 hybrid is crossed with so the F1 hybrid is going to be this, is crossed with a homozygous recessive brown. So that, okay, so this is going to give us this. Okay, yes, yeah, so it's going to be 1-to-1. So 1-to-1 for question 13. Then question 14 is an example of a test cross. That's it. Question 15. The genotype of a husband and a wife are, so we have a husband who is AB and the wife who is blood group O, or blood group A. O is recessive, so someone can only be blood group O if that individual has got homozygous trait like this. Okay, but when it is heterozygous, that individual is going to, in this case, this will be blood group A. Okay, then the question goes on to say, among the blood type of their children, how many different genotypes and phenotypes are possible? So here we just have to conduct a very simple cross. So we have got, I'm not going to use those arrows. So we have got A, B, being crossed with heterozygous O. So we're going to have A, B, then A and O. Okay, so this and that, we're going to have blood group A dominant, this and that, blood group A heterozygous, this one is going to be blood group AB, then this and that, this will be blood group O. So the question is, how many different genotypes? So we start with the different genotypes. So different genotypes, we have 1, 2, 3, 4. So 4 different genotypes. Against how many phenotypes? So phenotypes, we're going to have this individual be blood group A, this one also be blood group A. So we have 2 blood group A's, then this will be blood group AB, this will be blood group B. So we're going to have 3 phenotypes. So 4 genotypes and 3 phenotypes. Which one is that? 4 genotypes, 3 phenotypes. So the answer has to be option D for question 15. That's how simple you can answer this question. Question 16. Question 16 we have, you self-cross the F1 plants and observe the following ratio. So we have got 20 red, 42 pink, 18 white. Based on the probability value obtained using the calculated chi-square, p-value is greater than 0.5, or that is p-value is greater than 0.05. We conclude that, so you know to say that here, this question is based on the chi-square. So in the chi-square, you have the p-value. This p-value, you obtain it from the table. So if the p-value is greater than 0.05, what it means is that this p-value is greater than the chi-square. The chi-square, this is the value that you calculate. So when the p-value is greater than the chi-square, the results are accepted. That is, there is no significant difference between the observed and the expected values. Okay? So the results are going to be accepted. There will be no rejection. But if the things are to change, if the p-value now is less than, in that case it means that there is significant difference between the observed and expected values. So the hypothesis is going to be rejected. Okay? You are going to reject the results. So in this case, the question is saying the p-value is greater than 0.05. That is, it's greater than the chi-square. If it's greater than the chi-square, the results will be what? Accepted. There is no significant difference between the observed and the expected values. So A is saying the hypothesis cannot be accepted, so it will be accepted because it's greater than the calculated, which is the chi-square. B is saying the hypothesis can be accepted, yes. Then C is saying the genes are not associated independently, so they are associated independently. So for the fact that the p-value is greater than the calculated value, it means that the genes are associated independently. Then D is saying the genes must be linked. So the genes did not link because the p-value is greater than what? 0.05. But if it's less, then it means the genes did not associate independently and also the genes linked. So the answer here is the hypothesis can be accepted. So the answer will be option B. Okay, you are going to understand this much better when we get to section B. I think we have one particular question which is based on that, where we're going to explain some more details on how you handle the chi-square questions. 17. RNA polymerase binds to a dash to initiate transcription. So RNA polymerase binds to a region which is known as the promoter. Why is it going to bind there? Because it wants to initiate what? Transcription. So it wants to initiate transcription. So it has to be promoter transcription. So the answer has to be option B. Okay, we don't have in translation we don't have the promoter, as well as the regulator, no, DNA, no. So it has to bind to a region which is known as, this region is found on the DNA. So it's just a particular segment on the DNA which contains specific bases where RNA is going to bind. That's what we call a promoter. When it binds there, it's going to initiate what? It's going to initiate transcription. 18. Which of the following mutations is responsible for causing cystic fibrosis? So cystic fibrosis, this is a mutation where there's a deletion in a triplet bases are deleted. In short, a codon is deleted. Let's say A, C, G. These three bases are going to be deleted in the sequence. And these bases are deleted in a protein which codes for, in a protein which regulates water and salt entry into the cell. So it's going to result into mutation in that protein. So it's going to result into difficulties to regulate salt and water. So which of the following mutation is responsible for causing cystic fibrosis? So it's deletion, because a triplet base is deleted from the base sequence that controls the in a gene that controls the regulation of salt and water. So it's option A for question 18. The 19. Which gene or the like operon codes for the enzyme that allows lactose to penetrate into the cell? So we know that the lac operon has got 3 genes. It has got 3 genes. It has got lac, it has got lac Z, then it has got lac Y, and then it has got lac A. So lac Z codes for beta-glycosidase, which breaks down lactose into glucose and galactose. Lac Y, this one codes for a membrane-bound protein, which is known as penetrance. This is an enzyme which allows lactose from outside of the cell to get inside the bacteria cell. So that is a membrane-bound protein. Penetrance is encoded by gene lac Y. Lac A codes for an enzyme transacetase. So the answer there has to be lac Y. The lac operon codes for an enzyme that allows lactose to penetrate or to permeate into the cell. That is gene lac Y. So the name of the enzyme is penetrance. Okay. 20. A type of chromosome mutation that may arise as a result of synapses in prophase 1 is so we know that during prophase 1, the synapsis is the process by which the maternal and paternal chromosome come together gene to gene and align themselves gene to gene. So you have this maternal, you have another paternal chromosome. So these two chromosomes will come together. This one will come on top of this chromosome gene to gene to exchange some of their segment. So during this exchange, a mutation can happen. So which one amongst these chromosome mutation may arise as a result of synapses? So we have inversion, deletion, translocation, and duplication. So the highest possible mutation that can take place due to synapses is inversion. Inversion, you know to say that during synapses, there is exchange of segments. Let's say this portion is going to be cut and attach itself there. This one will be cut and it will come here. Okay. So during this exchange of these segments, it cuts. Then instead of it binding here the way it is, it can flip. So when it flips, that flipping is what is known as inversion type of mutation. So it will come from this one chromosome. Instead of it going direct the way it is, let's say this is part A, this is part B, then it flips now. It flips now, the B comes on top, the C comes down here. This is possible during synapses. So the answer there has to be what? Has to be option A. It can cut and flip, resulting to inversion type of mutation. So option A for question 20. Then 21. Polyploid refers to, so poly has to do with many. Ploid, those are chromosomes. So polyploid, you know that we have got pairs of chromosomes. Each chromosome exists as a pair. So when you say polyploid, instead of chromosomes occurring in pairs, they will be in sets of, let's say maybe 3, or some will be in sets of 4. That is what is known as a polyploid. You don't have a pair of chromosomes, you have more than a pair of chromosomes. In each set of chromosomes, those 23, instead of just being 2, 2, there will be, let's say, maybe 3, 3, 3, 3. All the 23 are going to be in pairs of, in groups of 3 instead of 2. That is, mutation is what is known as a polyploid. So it's saying polyploid refers to extra copies of gene adjacent to each other in a chromosome. B is saying translocation of one section of a chromosome to another. C is saying an individual with extra sets of chromosome. D is saying a chromosome which has replicated but has not divided. So the answer is going to be C. An individual with extra sets of chromosome. Extra sets of chromosome. Instead of these being in pairs, then they are in groups of 3. Each pair, chromosome 1, they are in groups of 3. Chromosome 2, groups of 3. Just like that, all the 23 pairs. That situation is what we call polyploid. Some can even be 4. Okay? That is polyploid. So an individual with extra sets of chromosome. 22. The genotype of an individual with Klinefelter syndrome is so an individual. This is going to be an individual, a male individual who has got XXY. you know that normal males have got a single X and a Y chromosome. But someone who has got Klinefelter has got two X chromosomes and one Y chromosome. So these individuals are going to be males, but they are going to possess physical characteristics of females. They are going to develop breasts, hips, they will not have beards and the like. Even their voice is going to be that of a lady. So these are going to be male individuals who have got an extra X chromosome. So you have got two X chromosome and a Y. This situation or condition is what we call Klinefelter syndrome. So the answer has to be option B. Two X chromosome and a Y. That individual is going to be male but with female characteristics. 23. Which of the following would modify the 3:1 Mendelian ratio to 2:1? So this can only be possible if one of the offspring dies in the 3:1. So let's say a particular cross that can give us this. Let's say if you are crossing this and what else? Which cross can give us where we are going to have something like this, then another one this, then the other one this. So you have got one or this, then two or these, and one or that. What should be? Okay, like this. Okay, so in this case, let's say an individual who is having this genotype. This is these two, if you are to follow the genotypes or the phenotypes, these three individuals are going to have what? The same phenotype. That is going to be 3 because we have 2 and 1, 3 to what? 3:1. Then let's say an individual who is carrying a recessive alleles. These recessive alleles are lethal. This individual will die. This is going to remain to be 3:0. Or an individual is carrying a dominant trait is going to die. In this case, it would mean that this one is going to die. This one, because it's heterozygous, is going to survive. So when this individual dies, because this is lethal, you're going to remain with 2:1. And that is what this question is telling us. It means that in this case, an individual carrying this trait, this genotype is going to die, only leaving us with 2 heterozygous and 1 recessive dominant, 1 homozygous recessive. So that has to be a lethal gene. So the answer has to be A. That is the only situation where you're going to modify that. That is one progene will die. So lethal gene, codominance, no. Incomplete dominance, duplicate gene, no. So it will only be possible, it can only be modified in that ratio if you have got a lethal gene. 24. What percentage of male offspring produced by a father with normal vision and a colorblind mother are expected to be colorblind? So this is an example of X-linked or sex-linked trait or disorder. So in this case, we are looking at a male want to find the percentage of male offsprings who will be produced from the family where the father has got normal vision. Colorblind is an example of recessive sex-linked trait. So let's say the father who is, this is going to be the father, then you have got the mother. So what is going to be the genotype of the father? The father with normal vision. So normal vision, let's use capital letter R, normal vision. So R, normal, then this color blind, this one blind vision, and a mother with a normal vision and a color blind mother. So for the mother to be color blind, she has to have this genotype. Okay, she has to have that genotype. So this is going to be the cross. So from this cross now we start. Because we're going to have 4 offsprings here, 2 sons and 2 daughters. So I'll start with this one. We have X, capital letter R, and that. So X, small letter R. That is going to be female, a carrier, normal but carrier. Then we also have this and that, the same genotype. this one, and that one. So it will also be female but a carrier. Then we come to this and that. So you start with X, this and that. This will be a male son, a son who is colorblind. And also this and that. We have another, this and that. So the question is, what is the percentage? The question is, what percentage of male offspring produced by the father who is normal for vision, and the colorblind mother, this one. So in this case, we are just focusing on the sons. How many sons do we have? We have 2. Or the 2, how many are going to be colorblind? So both of them are going to be colorblind. So we have 2 who are colorblind multiplied by what? Or 0 normal out of 2 multiplied by what? 100%. So this is going to be not 0. So not 0. Or the 2 sons, how many are colorblind? So we have 2, both of them are colorblind. Out of the total number of sons, 2 multiplied by 100%. So this is going to be 1 multiplied by 100, you have 100%. So no son in this family is going to have a normal vision. All the sons in this family are going to be colorblind. The reason being is because all the sons inherit the X chromosome from their mother. So each and every son is going to inherit the mutated X chromosome from their mother, thereby resulting to them being blind. For females, as you have seen, they can inherit the mutated X chromosome, but as long as there is a dominant chromosome, they are going to be normal but carriers, so the disease will not manifest in their phenotypes. They can be carriers. In short, in sex-linked disorders, females can be carriers, whereas males can only be 100% normal or 100% affected. It becomes worse when the mother is affected, especially if the disease is recessive. So the answer here is going to be D. All the sons in the family are going to be affected. They are going to be colorblind. 25. 2 parents, both heterozygous for blood group A. That is, if we say both are heterozygous for blood group A, it means they are like this. Okay? This parent is also having this genotype. Heterozygous for blood group A produce a child. What is the probability that the child has a type A blood group? So we just have to conduct a cross here. So I'll start. The first one is going to be this, dominant A, that is this and that. Then this and that, heterozygous A, okay? This is going to be heterozygous A, this and that. We have another heterozygous A. Then finally, we have this and that. This individual or this child is going to be blood group O. O can only be expressed if someone has got a homozygous genotype like this one. Is going to be blood group A or blood group O. This one blood group A because O is recessive. The same with this one. So the question is, 2 parents, both heterozygous for blood group A, produce a child. What is the probability that the child is blood group A? So we have 3 of the 4 are going to be blood group A. So we have 3 out of the total 4 multiplied by 100%. If you conduct your cross, this is going to be 4 into there, that is going to be, I think this will be 25. 25 by 3, that is 75. So 75 in decimal form, divide this by 100, it will be 0.75. So the answer has to be option C for this question. So this marks the end of section A. We now move on to section B.